The process of protein synthesis by Harry Coffey. Explain using lollies. The process of protein synthesis begins with a specific section of DNA. DNA is found in the nucleus of cells and made up of nucleotides, which have 4 different nitrogenous bases: adenine, thymine, cytosine, and guanine. These bases pair together through hydrogen bonds in complementary pairs. A pairs with T, C pairs with G. So adenine and thymine, and cytosine and guanine. Protein synthesis is made up of different processes. The first process is called transcription. In transcription, an enzyme called RNA polymerase begins to unzip the double helix of the DNA to expose one side called the template strand. The RNA polymerase, which in this case is an apple, unzips the DNA. When this happens, free nucleotides begin to copy the template strand in sequence. This process produces a single-stranded molecule called pre-mRNA. This is an exact copy of the template strand of DNA. However, RNA has a nucleotide base called uracil, which is used in place of thymine. Therefore, the base pairs are adenine, uracil, cytosine, and guanine. As the RNA polymerase continues to unzip the DNA strand, the pre-mRNA copies the entire sequence that it requires. Once the pre-mRNA sequence is complete, the original DNA is zipped back together, just like in the photo, unchanged as it was before. The pre-mRNA is now complete, but it needs to be altered before it can become mRNA. Therefore, the transcription process isn't yet complete. Pre-mRNA has sections called introns and exons. The introns do not code for anything in protein synthesis, whereas the exon sections are expressed. They are the parts in the sequence which are coded for. Because the pre-mRNA doesn't require its introns for protein synthesis, they are then removed in a process called post-transcriptional medication. This is carried out by a complex known as splitsosome. In this process, the introns are removed, but the exons are kept. Once this process has been complete, the original pre-mRNA strand becomes mRNA or messenger RNA. As I said, the remaining exon sections are binded together and create operational mRNA. This happens in a variety of different orders, which means one sequence can code for several different proteins. But we'll look at that in a little bit more detail a little bit later on. So once the pre-mRNA has been edited and becomes messenger RNA, it then leaves the nucleus through its pores and enters the cytoplasm of the cell. This is the end of the transcription process and the beginning of the translation process. Translation begins when the messenger RNA reaches the cytoplasm and attaches itself to a ribosome. A ribosome is an organelle responsible for the site of protein synthesis. In this case, it's a pumpkin. The ribosome read the mRNA sequence in groups of 3 bases. These are called codons. The ribosome recognizes the codons and brings a tRNA molecule containing a specific complementary base pairing sequence. This sequence is also 3 bases long and is located at the bottom of the tRNA molecule. It is called an anticodon. The codon and anticodons must match perfectly in sequence. Each tRNA molecule carries a specific amino acid which are the building blocks of proteins. In this example, the mRNA codon sequence is AUG. Therefore, the corresponding tRNA sequence must be UAC. The anticodon UAC codes for an amino acid called methionine. Methionine is the start codon. It will be the first of the amino acids in the chain. The next codon on the mRNA is a sequence of GAU, or guanine, adenine, and uracil. So the correct tRNA sequence required is CUA. This codes for the amino acid called aspartic acid. So the second amino acid in our protein sequence will be aspartic acid after methionine. As the entire sequence moves through the ribosome, more and more codons are read and more tRNA molecules are brought with the corresponding anticodons to bring more amino acids and build a polypeptide chain or a chain of amino acids. The sequence created from our lolly messenger RNA was methionine, aspartic acid, proline, and then it reached a stop codon, which meant that was the end of the polypeptide sequence. Now this photo was taken of 3 different amino acid sequences or proteins created from just one sequence of messenger RNA. But how is this photo possible? It's because of a process called alternative splicing, which is responsible for creating different proteins from the same mRNA sequence. So just to quickly recover it again, the pre-mRNA right from the beginning, which was copied from the DNA, has its introns removed at the end of transcription because they don't code for anything in protein synthesis. This leaves us with just the exons, or the actual coding parts of the sequence. But what alternative splicing does is it can leave out or change the order of the exons depending on the protein it would like to make. So therefore, there's 3 examples here. The first one in the top right corner, the first exon is left out, meaning the protein synthesis will only code for exon 2 and 3. In the second example, the second exon is left out, so it will only code for number 1 and 3. In the third example, exon 3 is left out, and the protein will only code for exon 1 and exon 2. Another example is it might only code for exon 1 or exon 2 or just exon 3. Because the sequences are different, they will end up with different proteins at the end or different amino acid chains, even though it's the same piece of messenger RNA to begin with. It's just been changed through alternative splicing. So now I'll show you how, on that photo before, the same mRNA sequence ended up with 3 different protein chains or 3 different sequences of amino acids. In this first example, we're going to remove an exon from the beginning of the sequence. So this first example, when we removed an exon at the beginning of the sequence, shows that the mRNA has a different starting point or codon sequence, and therefore the amino acid sequence will be different. Now, in protein synthesis, methionine is always the start codon, but to show you what happens, I'm going to remove the first exon. So the original exon produced methionine, but because it's been alternatively spliced and we removed that first exon from the mRNA, now our codon is UGG. Therefore, our anticodon on the tRNA needs to be ACC to match. This codon, UGG, codes for the amino acid tryptophan instead of methionine. Although the pictures are terrible, and I apologize for that, as I moved along the lolly sequence, you can see that the first sequence was methionine, aspartic acid, and proline. In the second sequence, because the first exon was removed, we ended up with tryptophan, serine, and leucine. Therefore, because there were different amino acids in the sequence, we've ended up with a different protein. A second example of alternate splicing is removing an exon from the beginning of the sequence, like we did in the first one, and the center of the sequence as well. We remove an exon from the middle. Because, like in the first example of alternate splicing, we removed the first exon, we have the same starting point in this example. So we have the codon UGG and the anticodon of ACC. So our first amino acid, will again be tryptophan. But because we removed the second exon, the next part of our sequence is different. And the next amino acid we code for is isoleucine. Therefore, as you can see, the top one is the first sequence we had that had all the exons involved. And then the two below are examples of what happens when we alternatively slice the exons, we leave some out or change them, and we end up with different protein sequences, amino acid sequences in proteins. But the important thing to remember is that they all came from the same mRNA sequence, which was copied from the original DNA template strand. The end. I'm going to go eat some lollies. Ew.